Some presynaptic terminals in the central nervous system appear non-functional, or silent. Here we present a protocol for identifying silent presynaptic terminals. This protocol can be applied to identifying treatments and manipulations that are suspected of silencing or awakening terminals. The protocol combines vital dye labeling of synaptic vesicles during neuronal depolarization with subsequent labeling of fixed cells with an antibody that labels all presynaptic terminals, both silent and active. Synapses co-labeled with dye and antibody are active. Synapses labeled only by antibody are silent.
Hi, I'm Amanda Taylor from the laboratory of Dr. Steve Mennerick in the Department of Psychiatry at Washington University School of Medicine.
I'm Xiaoping Jiang, also from the Mennerick lab.
Today we would like to show you a procedure for visualizing silent presynaptic terminals using cultured hippocampal neurons.
We use this procedure in our laboratory to study how electrical activity and other signaling pathways can modulate the neurotransmitter release.
So let's get started.
In a sterile laminar flow hood, use hippocampi from postnatal day 0 to 3 rats or mice to prepare dissociated cell cultures according to our published protocol. Our cultures are mixed neuronal glial cultures derived from the same dissection. Cells should be plated in sterile 35 mm dishes, each containing a number 0 cover slip that was coated with 5 mg per milliliter of collagen. Plate the neurons at an approximate density of 500 cells per centimeter squared. 12 rat pups will typically yield 30-35